the plants make its own food material by using of the raw materials like carbohydrates, water, sunlight, chlorophyll, pigment.
Right.
And many other processes are also involved in the photosynthesis 1 and 2.
Right. Is it catabolic process or anabolic process?
It is an anabolic process.
Anabolic means synthesized process. So how many stages in photosynthesis?
Anabolic.
Right. How many stages?
3 stages, sir.
What are those? As the definition, synthesis of food material, right? I'm asking you how the food material synthesized.
Light and dark reaction, sir.
Light and dark reaction, okay. Which is first, light or dark?
So first is the light reaction, light dependent reaction. Next is
light dependent reaction. What is the end product of light dependent reaction?
NADPH plus ATP, sir.
NADPH, right? ATP.
Yes,
and
sir.
this energy called as assimilatory power, right?
Yes, sir.
So mainly this is carried out inside the thylakoid membrane. The light reaction takes place in thylakoid membrane, right?
Yes, sir.
The end product ATP NADPH, it is utilized in what is the further reaction?
Dark reaction.
Dark reaction. So what is the end product of dark reaction?
Starch, sir. C6H12.
Starch formed inside the in which region starch produced?
Thylakoid, sir.
In which region of chloroplast starch synthesized?
Stroma,
in
sir.
chloroplast, yes, in stroma.
Stroma.
So in stroma, the starch material synthesized by C3 cycle or C4 cycle or CAM cycle, right?
Yes,
so,
sir.
That's what we learned in photosynthesis. After formation food material in stroma region, the next step is, next step would be after formation, after synthesized food material, next step, it undergoes hello?
Yes. Yes, sir. What is your question, sir?
Just wait. I was curious. Right. Please be silent. Right. What is my last question? So, what happened after formation, food material in stroma? What would be the next step? After formation, food material, next.
Which will transfer to all the parts of the plant, sir.
Okay, next.
Utilization of the food material.
Okay, next. How it is utilized?
How it is utilized?
For respiration.
In the form of growth and repair.
Transportation, sir.
What is transportation? Okay. After transportation, it is settled at consuming plants, consuming parts, such as fruits and leaves, and flowers, whatever, and roots also. But so after utilizing, so it is utilized in the form of the food material utilized in the form of
Carbohydrates, sir.
the carbohydrate formed in storage part only. The carbohydrate and starch are storage food material. That is the excess of carbohydrate storage in plant part.
Yes, sir.
But before that, respiration takes place. After formation food material, respiration takes place.
Okay.
Right?
Okay.
What is respiration? What is respiration? Simple question.
Breaking down
Exchange
of
of
glucose,
Breaking
Breaking
down of
carbon
the
down.
glucose.
dioxide, and energy
Right.
along with water.
Right. Okay. It's clear. Then how many stages in respiration?
2 stages, sir. Aerobic and anaerobic.
What is aerobic?
In the
With the
presence
help
of
of oxygen, in the presence of oxygen, respiration takes place, sir.
Are you able to see the board?
yes.
Yes, sir.
Right. Is it clear?
Yes, sir.
Right.
Yes, sir.
So as you said, aerobic respiration, aerobic and anaerobic. Right? What is aerobic? What is the end product of aerobic?
Carbon dioxide, water,
Yes,
and
water.
energy.
I'm asking. Okay.
Yes, sir.
Just give me the simple.
ATP
Right.
molecules production, sir.
ATP molecule production. How the ATP molecules are produced?
With the help of some enzymes involved in respiration.
These enzymes react on
This enzyme
what is
react on the substrate molecule.
right? What is the first substrate for respiration?
I think pyruvic acid, I think.
What is the end product of photosynthesis?
Glucose.
Glucose, sir.
Oxygen.
Right.
Glucose.
So the glucose would be the substrate of the respiration.
Yes, sir.
Right?
Yes, sir.
This is substrate for respiration by the aerobic respiration or anaerobic respiration. Right? First, in respiration, first step, only glycolysis takes place. What is that? Glycolysis. Right? So that is the first stage of respiration. Glycolysis. Okay. In aerobic, see this glycolysis respiration is the first step for aerobic and anaerobic. What is the end product of glycolysis?
Pyruvic acid. Pyruvic acid.
Pyruvic acid. Pyruvic acid, end product of glycolysis. But now the utility of the oxygen, that is utility of oxygen of pyruvic acid, it is like divided aerobic and anaerobic. If it is utilized oxygen, that is aerobic. Without oxygen, that is anaerobic. First, let's discuss about aerobic respiration. After that, anaerobic respiration. Okay?
Okay, sir.
Okay, sir.
Right.
Okay, sir.
First, in aerobic respiration, first step, already told you, the first step is
Glycolysis.
glycolysis. Glycolysis, that is the first one. So, end product glycolysis, as you said, that is the pyruvic acid, right?
Pyruvic acid.
2 pyruvic
After
acid
that,
molecules.
yes. After that, one connective link. The second stage is oxidative decarboxylation. Oxidative decarboxylation. Decarboxylation. What is third one?
Krebs cycle.
Right. Krebs cycle. And fourth one?
Electron transport.
Electron transport system. Right. Fourth one, electron transport system. So as you said, the end product of respiration, ATP. The ATPs are formed after electron transport system. After electron transport system, the number of ATPs are formed. This ATP energy utilized for bioactivity of cells, bioactivity of organisms. Right? Now let's discuss one by one. First of all, the glycolysis. Right? Before that, in general, as you said, the respiration is the catabolic process. Photosynthesis is the anabolic process. And it is not only catabolic process and exergonic process. That means heat generated during respiration process. So I'm asking, I would like to ask a simple question. What reason for absence of respiratory organ in plants? Does it have any respiratory organ in plants?
Tomato and lenticels.
I am asking about respiratory organ, special respiration organ or organ system.
Mitochondria.
Respiration system.
Mitochondria.
No, sir.
You are unable
No,
to understand my question. Listen
There is no
carefully.
special organ system, sir.
Right. What is the reason?
Tissue.
What is the reason? Any
So
animal have the special respiratory organ and respiratory system also.
They belongs to eukaryotes, sir.
Eukaryote. What about animals?
Animals also eukaryotes.
Right. What is the difference?
Plants.
Simple. Each plant part can take care of its own gas exchange.
Through stomata.
Yes. This stomata, whatever it may be, as you said, stomata, lenticels, they are almost all present throughout the plant body. Can our skin exchange the gases? Hello?
No.
No, sir.
No. But here, almost all plant cells can exchange. This tend to be exchange the gases. And there is a little transport of gases from one plant part to another. No need to system to transfer one place to another place. And plant do not present the great demand for gas exchange. Here the roots, stem and leaves can respire at rates, whatever it may be, maximum level of respiration. And when compared with animal, the rate of respiration lower when compared with animals. That is the only one reason here the gases exchange takes place inside the cell. That means almost all cells involved in gases exchange. That's why the no need to develop any special organ. Right? Now let's discuss the first stages, that is the glycolysis. Who discovered glycolysis? Let's share that short, just wait.
Porter, sir. Is it Porter?
Oh, no.
Is it Meyerhoff, sir?
Right. Mayer. Emden Mayer of pathway. Another name, Emden Mayer of pathway. Just
EMP
let's
pathway.
see. Right. Are you able to see this?
Yes, sir.
Is it clear?
It's very small, sir. You need to zoom in a little bit. So could you adjust it for full screen, sir?
Just wait. Hello.
Sir.
Yes, sir.
Oh, sorry for the disturbance. Just wait. Is it okay?
Yes,
Hello?
sir, it's clear.
Right.
Yes, sir.
Right. So first it is discovered by Emden Meyer. So another name called as EMP pathway. Remember it? EMP pathway, right? The glycolysis mainly occurs in cytosol, not a cytoplasm. Occurs in cytosol, remember. Cytosol. The photosynthesis end product, carbohydrates formed in, for example, this is the cell. Are you able to see this right line, red line?
Yes,
Hello?
sir.
Right. For example, this is the cell, red line cell. The green color chloroplast and this yellow foldings mitochondria. Okay? Is it clear?
Yes,
Okay,
sir.
sir.
Right. So first the photosynthesis takes place inside the chloroplast and food material formed, F for food material formed in chloroplast. Before respiration, this prepared food material diffused out. Diffused out. What is the, in between the chloroplast and mitochondria, this place called as,
Cytoplasm.
that's why don't get confused. I already said that is not a cytoplasm. That is the
Cytosol.
cytosol. What is the difference between cytoplasm and cytosol? And protoplasm and protoplast? Frequently getting confused with those words. The simple differentiation, the protoplasm means except the cell wall and plasma membrane, the entire content of the cell called as protoplasm. Cytoplasm without nucleus. With nucleus, protoplasm.
Yes,
Without nucleus, the entire content.
cytoplasm.
Cytoplasm. Without nucleus means another rest of the cell organelles also present. Mitochondria, Golgi complex, endoplasmic reticulum, those are all. But in cytosol, no cell organelle. Only liquid portion, no cell organelle. That's why this between the mitochondria and chloroplast, this liquid space called as cytosol. Now the glucose diffused out, settled here. This region called as cytosol. Now the respiration start from this region. In glycolysis, the partial oxidation of glucose takes place and little amount of ATP also produced. Right? What is the substrate? Already told you here the substrate itself is the glucose. Glucose is the substrate. Already the carbohydrate formed in chloroplast and diffused in the form of glucose, or it may be sucrose also. Maybe glucose or sucrose or any other sugar forms. The first step, phosphorylation takes place. First step called as this stage, phosphorylation. This reaction called as phosphorylation. Phosphorylation. What happens in phosphorylation? Glucose substrate with help of exokinase enzyme. This enzyme, exokinase enzyme, ATP, one phosphate loss. This phosphate received by, this phosphate T means triphosphate, one phosphate loss, and this losing phosphate received by glucose. So here only glucose. After received phosphate, it converted into, hello? It
Yes, sir,
converted
we can
into,
hear.
Yes, sir.
converted into first only the simple glucose with add one phosphate from ATP. That means adenosine triphosphate. One phosphate received and convert into glucose
Glucose 6-phosphate.
6-phosphate. Glucose 6-phosphate. Now the tri becomes diphosphate, ADP. Right? So here
Yes, sir.
this reaction carried out by exokinase enzyme. Now second reaction, isomerization. Isomerization. So here the involved enzyme is phosphoglucose isomerase. The isomerase only internal change of the atoms. Internal change. That is glucose 6-phosphate converted into fructose 6-phosphate. What is the difference glucose 6-phosphate and fructose 6-phosphate in structurally?
Glucose has a CHO group, sir. Fructose is having COOH group. Acetone group. In glucose, aldose group is there.
Glucose fructose difference?
They are isomers of each other, sir. But the
before
functional
that,
group is different.
Before that, what is the formula of glucose?
C6H6O12.
C6H12O6.
What about fructose?
C6H12O6, sir.
Same, sir.
What about same?
Same.
But these both are mirror images.
Yes, sir. Isomers to each other.
Right, isomerase. That's why it is called as isomerization reaction. The involved enzyme is isomerase. Now, the third reaction again, phosphorylation. So first phosphorylation, second isomerization, and third one phosphorylation. Let me draw here. Same phosphorylation. Don't make sound, please. Hello. Thank you. Right. During phosphorylation, the same year, like a glucose, convert to glucose 6-phosphate. The fructose 6-phosphate, again add another extra phosphate converted into fructose 1,6-biphosphate. The same reaction carried out here. Like a first reaction, ATP triphosphate, donate 1 phosphate. This phosphate received by fructose 6-phosphate and it is converted into ATP to ADP. As a result, the fructose 6-phosphate converted to fructose 1,6-biphosphate or bisphosphate. The same enzyme is kinase. The first enzyme also kinase, exose kinase. Here also same kinase enzyme involved in this reaction. Right? In fourth reaction, cleavage. Fourth reaction here, cleavage. Please write down this cycle. Cleavage. What will happen in cleavage? The 6-carbon fructose 1,6-bisphosphate cleaved into 3-carbon glyceraldehyde, 3-carbon glyceraldehyde adenosine phosphate, GLAP, and dihydroxyacetone phosphate. These both are 3-carbon compounds. 3-carbon. Here also, this glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate also 3-carbon. So the enzyme is triose phosphate isomerase. Why are the only isomerase involved? Before that, aldolase enzyme involved. After involved isomerase enzyme. Aldolase enzyme involved for only cleave of the fructose 1,6-bisphosphate. With alpha of aldolase enzyme, the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate both are formed. Next, they can interchangeable like fructose and glucose. That's why this reaction also considered as a isomerization. Are you clear? Hello?
Yes, sir.
In glucose 6-phosphate convert into fructose 6-phosphate, isomerization reactions involved the same way. Glyceraldehyde 3-phosphate, DHEAP, dihydroxyacetone phosphate also isomerization reaction involved isomerase enzyme. It means these also these mirror images like glucose and fructose. Right? So isomerization, fifth one. Now sixth reaction. Sixth one, dehydrogenation. This is called as a oxidation process. Oxidation. Loss of hydrogen. Loss of hydrogen considered as a oxidation, right? So here this oxidation process. Oxidation process. What will happen here? glyceraldehyde 3-phosphate convert into 1,3-bis phosphoglyceric acid. Better to write down this cycle. Right. Here NADP or NADP converted into NADH2. Converted into NADH2. That means from the glyceraldehyde 3-phosphate, 2 hydrogens and as a result converted into NADH. Right? So this reaction called as oxidation reaction. The first actually not two, only one NADH form. And next step, after formation 1,3-bis phosphoglycerate, same again phosphorylation takes place. Before that, how many phosphorylation takes place here?
2, sir.
2. So here the third one. The phosphorylation takes place here. So it is this phosphorylation called as substrate level phosphorylation. I will tell you what is substrate level phosphorylation. So after formation 1,3-bis or biphosphoglycerate through the oxidation process, by substrate level phosphorylation, 3-phosphoglycerate form. 3-phosphoglycerate. So, but in this phosphorylation, ADP, diphosphate, not triphosphate, diphosphate converted into triphosphate. That means what would happen here? Hello?
1 phosphate molecule is added, sir.
From where?
From 1,3
A,
glycerol.
Right. From 1,3 biphosphoglycerate, 1 phosphate atom lost, 1 phosphate lost, and it received by D. Adenosine diphosphate, as it received another phosphate, it is converted into triphosphate.
yes, sir.
So here the phosphorylation takes place in substrate. That's why it is called as substrate level phosphorylation. That means here the phosphate released from the substrate. That's why it is called as substrate level phosphorylation. So 1 ATP formed. But here given 2, I will explain later how the 2 formation. Right after formation, 3-phosphoglycerate with help of mutase enzyme, 2-phosphoglycerate form. 2-phosphoglycerate. Here in this reaction, what will happen? Just the phosphate shifted to 3rd carbon to 2nd carbon. Simple. So that's why it's called as a 2-phosphoglycerate. Same first 3-phosphoglycerate. As it shifted to 2nd carbon, it is considered as a 2-phosphoglycerate. Next one, with help of enolase enzyme. In enolase, what will happen? The water molecules received. Water molecule received, as a result, phosphoenol pyruvate. Phosphoenol pyruvate formed. Phosphoenol pyruvate formed. Next, again this pyruvate kinase. The same way ADP, ATP converted into ATP and pyruvate enol phosphate converted into pyruvate or pyruvic acid. Is it phosphorylation reaction or substrate level phosphorylation? Hello?
Sir,
The last
substrate level
reaction.
phosphorus.
Right. So how many phosphorylation takes place in glycolysis?
In the first step, sir, before cleavage, 2 phosphorylation, sir. After that, 2 phosphorylation takes place.
I am asking only phosphorylation.
Only phosphorylation 2, sir.
And what about substrate level phosphorylation?
2, sir.
That's it. Just wait. So actually we completed this cycle from glyceraldehyde 3-phosphate only. Just see this red color. What is remain? What is that?
Dihydroxyacetone phosphate, sir.
Right. This dihydroxyacetone phosphate, after formation 1 pyruvate, Again, this dihydroxyacetone phosphate converted into glycerol 3-phosphate through the isomerization reaction.
Okay, sir.
Be careful, right? So after formation glycerol 3-phosphate, the rest of the reactions the same. That means the first reaction would be oxidation, formation ATP, another NADPH. That's why it is the 2 NADH. First from the glycerol 3-phosphate 1 NADH. The second after converting DHEAP into glycerol 3-phosphate again undergoes to oxidation. That's why another NADH form. The same way, first 1 ATP form, the next cycle another ATP, 2 ATP. That's why 1 ATPs are added. That means here from the glycerol 3-phosphate, the cycles run 2 times. One is from direct glycerol 3-phosphate, another cycle from when convert the dihydroxyacetone phosphate to glycerol 3-phosphate. Now tell me how many substrate level phosphorylation takes place in glycolysis?
4, sir.
Right. 4. How many oxidation cycles takes place in glycolysis?
2, sir.
2. How many NADH2 are formed in glycolysis?
How many? 4, sir.
2.
2
only.
sir.
Every time 1 NADH only.
Okay, sir.
So 2. How many ATPs are formed in glycolysis? This is very, very important.
4 ATP, sir.
4.
4 ATP. Right?
But 4 ATP, sir. Before that, 2 are already utilized. So remaining net gain is 2 ATP, sir.
That's it. So actually total 4, as it already previously utilized 2 ATP, the net ATP number 2. Okay? Write down on the notes. Please write down. Because it is utilized for balancing sheet of ATP. Glycolysis, 2 ATP. Okay? Clear?
Yes, sir.
How many NADH2?
2 NADH.
Write down. Okay? Clear? Right. In aerobic, next step. What is the next stage?
Krebs cycle, sir.
Before Krebs cycle, already given another method also.
Oxidative decarboxylation, sir.
Pentose phosphate pathway.
Pyruvate decarboxylation pathway. Just wait. Second step in aerobic. In aerobic, second step is pyruvate decarboxylation or oxidative decarboxylation. This. So end product of glycolysis 2 pyruvate, right?
Yes, sir.
Right. So the pyruvate number 2. 2 pyruvate. So finally, it is converted into acetyl coenzyme A, 2 acetyl coenzyme A, 2 acetyl coenzyme A, right?
Yes, sir.
Here, where is glycolysis takes place?
Cytosol.
Right. So in pyruvate formation also in cytosol only. After formation of pyruvate in cytosol, this glycolysis pyruvate transported to mitochondria. Right?
Okay, sir.
Mitochondria. It is carried out by pyruvate translocator that catalyze an electroneutral exchange of pyruvate and OH minus across the inner membrane. So as a result, now this pyruvic acid or pyruvate enter into the mitochondria. Now the further cycle takes place inside mitochondria. The first reaction, first respiration reaction in cytosol, the further reaction all in mitochondria. So let's see the oxidative decarboxylation or pyruvate decarboxylation. Why is oxidative decarboxylation means? Here, the first step decarboxylation takes place. Decarboxylation. First step decarboxylation. And second step, oxidation. And third one, conjugation. Third one, conjugation. Mainly 3 stages involved in this oxidative decarboxylation. Right? First, decarboxylation. Decarboxylation means loss of carbon dioxide. The loss of carbon dioxide. Loss of carbon dioxide. Hello, please. Okay, thank you. So loss of carbon dioxide is called as a carboxylation. Carboxylation. Next one, oxidation. What will happen in oxidation? NAD converted into NADH2. Converted into NADH2. That means this NAD receive hydrogen and converted into NADH2. That is second reaction. That's what it is called oxidation. And next one, conjugation. Conjugate means fuse with coenzyme A. Coenzyme A. Fuse with coenzyme A. As a result, C keto groups here. Coenzyme A as it is. CH3 as it is. Now this is called as a acetyl coenzyme A. So here the 2 pyruvate converted into 2 acetyl coenzyme A. That's why here the 2 NADP converted into 2 NADH2, right? Hello?
Yes, sir.
Yes, sir.
So how many NADPH2 form? How many NADH2 are formed in oxidative decarboxylation?
2, sir. 2.
2. What about glycolysis?
Again 2, sir.
2, sir.
2 only, sir.
Write down this both. Record it. So in oxidative decarboxylation, 2 NADH2 are formed. How many ATP numbers?
ATP.
Please be silent. Hello. Please be silent. How many ATPs are formed in oxidative decarboxylation? Is there any ATP?
No, sir.
No, sir.
No ATP formation, right? Only NAD converted into NADH2. That's why 2 NADH2s are formed. Right? Actually, pyruvate, right? Remember, here the pyruvate carbon number. How many carbons in pyruvate?
3.
3 carbons.
3 carbons.
3.
1, 2, and 3 carbon. What about acetyl coenzyme A?
2.
It's
2,
2
sir.
carbons, sir. 2 carbon
2
molecule.
carbons. Yes, already in the form of carbon dioxide, 1 carbon lost.
Decarboxylation.
Right. So 2 carbon acetyl coenzyme A, end product of oxidative decarboxylation. Is it clear?
Yes, sir.
Yes, sir.
Right. Here the transport of pyruvate into the mitochondria, already told you, by pyruvate translocator. It transports pyruvate in a symport fusion with a proton. It is the active basis consuming energy. That means for after formation pyruvate, for transport into the mitochondria, it utilized some energy. Right? Next one. What is the next cycle?
Krebs cycle.
Krebs cycle,
So
sir.
remember the end product acetyl coenzyme A. Now the cycle start with acetyl coenzyme A, right?
Yes, sir.
Right.
Yes, sir.
Krebs. Who discovered Krebs cycle or citric
Sir
acid
Hans
cycle?
Krebs. Sir. Sir
Already
Hans
name
Krebs.
itself Krebs. Right. He got a Nobel Prize for discover this cycle. And another name, citric acid cycle. Is it clear? Just wait. Right. Citric acid cycle, and this Krebs cycle takes place in
Mitochondria.
as mitochondria matrix. Takes place inside the mitochondria matrix. Right. Let's see one by one. Whoa. Is it clear? Are you able to see?
Yes, sir.
Yes, sir.
Krebs tricarboxylation acid cycle or citric acid cycle. Right. This is acetyl coenzyme A, end product of oxidative decarbylation, right?
Yes, sir.
Okay. Right. This acetyl coenzyme A condensation with oxaloacetate, which is 4 carbon compound. 4 carbon compound. So oxaloacetate 4 carbon compound. This way. Oxalo state, 4 carbon compound, 1, 2, 3, 4. Now, what about the acetyl coenzyme? How many carbon?
2 carbons.
Only 2 carbon, right? So, as a result, formation of citrate or citric acid. Now, the number of carbon, citric acid,
6 carbons.
6 in citrate or citric acid, 6 carbon formed. So, condensed. Here, the condensed means attached the acetyl coenzyme A to the oxaloacetate or oxaloacetic acid. So here the enzyme involved synthase, citrate synthase. Enzyme is citrate synthase. Next one, after formation citrate, isocitric acid form. Isocitric acid form. Later, citric acid. After that, actually it must be here.
Oxoglutarate, sir.
Cis-aconitic acid also. Next, isocitrate. The first from the citric acid to cis-aconitic acid by loss of water molecule. From citric acid, loss of water molecule, cis-aconitic acid formed. And again, the gain of water molecule, isocitrate formed. Right? After formation isocitrate also 6 carbon. Don't get confused. Right? After formation isocitrate, it is next enter into the oxidation. It is the first oxidation. In this NAD convert into NADH2. Remember, NAD convert into NADH2. And the enzyme is dehydrogenase because the hydrogen lost from this isocitrate and this hydrogen received by NAD. It's converted into NADH2. As this hydrogen, as it lost from the isocitrate, it is considered as a dehydrogenation. Here the enzyme is dehydrogenase. Enzyme is dehydrogenase. Actually, as a result, here the formation of before alpha ketoglutarate, oxalosuccinic acid form. After formation oxalosuccinic acid, decarboxylation takes place. This is the second step. Decarboxylation, second step. Remember, write down before formation alpha ketoglutarate, oxalosuccinic acid form. Let me draw here, right here. Just wait. After isocitrate, oxalosuccinate formed by only oxidation process. From the oxalosuccinate, one carbon molecule loss. That is called decarboxylation. Right? After decarboxylation, alpha ketoglutarate acid form. Alpha ketoglutarate or alpha ketoglutarate acid form. Now how many carbon numbers in alpha ketoglutarate acid?
5 carbons.
Right. Because here already one carbon dioxide loss, that's why it is 5 carbon only. Right, after formation alpha ketoglutarate acid, now it is enter into the another oxidation and another decarboxylation. So already in this oxidation is the second one, right? This is the first
Yes,
one
sir.
oxidation. This is second oxidation. In second oxidational same NAD received hydrogen from the alpha ketoglutarate as a result formation of NADH2, right?
Yes, sir.
As a result formation of succinyl coenzyme A. This is succinyl coenzyme A. Write down succinyl coenzyme A. The how many carbons in succinyl coenzyme A?
4, sir.
Four. No,
Five.
no, no, no.
4 plus coenzyme A.
Four.
Four, sir. Four.
Four, because here already another carbon dioxide also lost. Next, you add coenzyme A. That's why next attach the coenzyme A. That's why it is called succinyl coenzyme A. Right? After formation succinyl coenzyme A, with help of GDP, utilize phosphate formation GTP. But remember here it is not a GTP. Write down ATP.
ADP.
ADP converted into ATP. Remember in plant cell respiration, ATP in Krebs cycle, ATP form. Whereas in animal cell, GTP form. Okay. Here given GTP, but you correct it ATP. GTP also right, but it is not in plant cell. It is animal cell. In plant cell, ATP formed. Right? ADP, one ATP formed as a result formation of succinic acid or succinate. We can see it. Succinic acid, succinate, right? After formation succinate, another oxidation, that is third oxidation takes place. In third oxidation, this succinate or succinic acid converted into fumaric acid or fumarate. Succinic acid converted into fumarate. So in this oxidation, instead of NADH, FAD converted into FADH2. FAD converted into FADH2 in every oxidation process from the substrate, hydrogen loss, this losing hydrogen attached to NAD converted into NADH2. The same way here also, from the substrate, hydrogen lost and attached to FAD converted into FADH2.
Yes, sir.
Before two oxidation, NADH2 form, but in third oxidation, FADH2 form instead of
FADH2.
NADH2.
Yes, sir.
Here the enzyme is succinate dehydrogenase enzyme. So after formation fumarate, fumaric acid, with help of fumarase enzyme, malic acid formed or malate formed. Malate or malic acid formed. Fumarase, that means in this reaction, water added. Water molecule added, as a result formation of malate. After formation malate and fourth oxidation takes place. Fourth oxidation takes place. In this oxidation also, always remember the oxidation enzyme dehydrogenase, removing hydrogen enzyme. That's why dehydrogenase, called as dehydrogenase. Sorry. Dehydrogenase. So the finally formation oxaloacetic acid. From the malic acid, after oxidation process, oxaloacetic acid formed. This cycle start with oxaloacetic acid combined with acetyl coenzyme A and end with formation of oxaloacetic acid, 4-carbon acetic acid. This is one turn of Krebs cycle. For one acetyl CoA. But how many acetyl coenzyme A formed?
2, sir.
2. So how many cycles would be run?
2 cycles.
2 cycles. Remember. The first of all, I would like to ask one simple question. How many oxidation takes place in
3,
Krebs
sir.
cycle?
4.
4, 4.
4.
Sure.
Yes, sir. 4.
Yes, sir.
Clear?
Yes, sir.
How many
Sir, in one.
ATPs? Ah,
Sir, are you asking for 1 cycle or 2 cycles, sir?
Krebs cycle means always remember, always it is 2 cycle, 2 turns.
Okay,
Cycle
sir.
1, but it is 2 turns,
Okay, sir.
right?
Yes, sir.
Now tell me, how many oxidations? Okay. How many NADH2 are formed?
6
sir.
NADH2.
If considered
sir.
2, 6.
6
6
NADH2, sir.
Right. 6 NADH2 are formed. Write down 6 NADH2. Glycolysis of NADH2.
Yes, sir.
2 NADH2.
Sir, 2
Glycolysis.
FADH2.
Right. In oxidative decarboxylation. In oxidation.
2, sir. 2.
2 NADH2. What about in the Krebs cycle?
6,
Right.
sir.
2.
How many FADH2 in Krebs cycle?
2 FADH2, sir.
2, sir.
How many ATPs are formed in Krebs cycle?
2.
2. So direct ATP in glycolysis.
Direct
Glycolysis ATP number.
4,
That
sir.
means total
4, sir.
total
Direct.
ATP. Direct means
4.
net ATP number.
Net ATP 2
4.
ATP, sir. 2
What about
ATP.
oxidative? Right, okay. Oxidative decarboxylation ATP number.
ATP number 0.
Right. Krebs cycle ATP number.
2 ATP.
Right, enough. Is clear?
Yes, sir.
So far, how many NADH2 are formed?
Overall 3 cycles, sir.
Yes.
6 plus 2,
10.
8. 8 plus 9, sir. 9, 10, sir.
10
10.
NADH2. How
10,
many?
sir.
Right. How many FADH2 formed?
2 FADH2.
Total ATPs?
2, sir.
2, sir.
Total ATPs? The
2,
all
sir.
cycles.
4.
Glycolysis. Right, 4. Write down these are all. Okay?
Yes, sir.
Right. Now electron transport system. Before electron transport system, just wait. What happened in electron transport system? The main concept of electron transport here. NADH2 oxidized into ATP formation. NADH2 oxidized into ATP formation. This is the main concept. So remember, is it okay first? What happened in electron transport system? That means this NADH2 oxidized. NADH2 oxidized means released electron. This electron transported through the crystal membrane and finally formation ATP. I will explain later clearly. But first just develop the concept. So if NADH2 oxidation, ATP formed. Is it clear?
Yes, sir.
Right. So far, how many NADH2 formed?
Total 10
Just wait.
NADH2, sir.
Just wait. Let me write here. glycolysis, oxidative decarboxylation, Krebs. Here 2 NADH2, right? Here 2 NADH2, right? Here 6 NADH2, right?
Total 10.
Right? Clear?
Yes, sir.
And
Yes,
another
sir.
oxidative reagent here, the FADH2. How many? 2 FADH2. What is our concept here? NADH2 finally produces the ATP. Remember, one NADH2 except glycolysis or except FADH2. In oxidative decarboxylation and Krebs cycle, one NADH2, if undergoes oxidation process, from one NADH2, 3 ATPs are formed. 3 ATPs are formed from one NADH2. Remember.
Except the
But,
NADPH of glycolysis, sir.
oh, no.
Okay.
Let me complete first.
Okay, sir.
In glycolysis, one NADH2 undergoes oxidation process, 2 ATP formed. Remember, don't get confused. I will explain clearly. First, just develop concept. One NADH2 of glycolysis enter into the electron transport system, finally produces 2 ATP. Okay? Is it clear?
Yes, sir.
In oxidative decarboxylation NADH2 and Krebs cycle NADH2, if these two type of NADH2, if enter into the oxidation process, for NADH2 oxidation, 3 ATPs are formed. 3 ATPs. Here also 3 ATPs. For one NADH2, 3 ATP. Here the FADH2 also, one FADH2, 2 ATP formed.
So that means, sir, if there are 2 NADPH2, so we have to multiply by 2 again, sir. 3
Exactly.
ATP. So it becomes 6 ATP.
Exactly. Now I'm asking one by one. First of all, glycolysis NADH2. If glycolysis NADH2 undergoes to electron transport system, how many ATPs are produced?
2 ATPs
6.
are formed, sir.
2 ATPs for how many
For
NADH2?
one NADH2.
Right. How many NADH2 are formed in glycolysis?
2, sir. 2 NADPH.
2 NADPs,
So
right?
total 4 ATPs.
Right. That's enough. Let me change the color. Total 4 ATP. Okay. Right? Is it clear?
Okay, sir.
Now,
Okay,
what
sir.
about oxidative decarboxylation reaction for NADH2? But here instead of 2 ATP, 3 ATPs are formed.
3 ATPs are formed. So total 6 ATPs are formed in oxidative decarboxylation, sir.
Right. 6 ATP formed. Clear. What about in Krebs cycle?
6
3
NADH2.
ATP. 6.
6 NADH2. 6 3
So
18.
18 ATPs
18
are formed.
ATPs
Sir.
are produced.
18 ATPs are formed. Right? What about FADH2?
One
2
FADH2
are present.
produces 2 ATPs, sir.
For one FADH2. But in Krebs
One
cycle, how many?
So
But in
total
Krebs,
2
4.
FADH2
2
are there, sir. So
FADH2,
4
sir.
ATP.
4 ATP.
4 ATP.
Now here, how many total ATP in glycolysis? 4, right? And decarboxylation 6, right?
Yes, sir.
And glycolysis
Krebs.
18, right?
Ha, sir.
And from 4 from
FADH2.
FADH2.
Ha,
32.
yes, sir.
Okay. How many?
32.
28, 32.
But if glucose undergoes to oxidation, how many ATPs are formed?
32.
In book, how many ATP is given? 36 ATP. So
Yes, sir, 36.
these 4 ATPs, where are these 4 ATPs? Where comes from?
They are from phosphorylation, sir.
Right. Already told you glycolysis 2 in Krebs cycle 2, right?
Yes, sir.
Total
They are
430.
direct ATP, sir.
Right, they are the direct ATPs. So total number of ATPs, if one glucose molecule undergoes to oxidation, total 36 ATPs are formed. 36 ATP formed. But in book of Tej and Jaigar, somewhat different. Tej and Jaigar given somewhat different. Actually, in some books, it is given as a one glycolysis, one NADH2 of glycolysis, 2 ATP produced. But in Tej and Jaigar book, given as a 1.5, not 2 completely. 1.5 ATP formed from one NADH2. Here
Yes, sir.
in Krebs cycle also not completely 3, 2.5 ATPs are formed.
Sir, that is the reason they are getting total 30.
Yes. What about our book? How many? I did not see this.
It is also mentioned 30, sir, in the book. ATP per glucose molecule, total they have given 30. Like you have told us, sir, FADH2 they have taken 1.5, NADH2 they have taken 2.5.
Right, let me see here.
So page number 235 I have.
235.
Yes, sir.
Right. Right. Here clearly balancing sheet clearly given instead of 2 ATP 1.5. So 2.5, 2.5, 1.5. If you mentioned 2 or 3 also right, or 1.5, 2.5 also right, you should follow any one. Either 3 or 2.5 as you wish. Because already by my choice is better to follow our book, our university book. And also the most famous book, Tej and Jaigar, also 1.5, but not completely 2. So the total number of ATPs is 30. Because the most of the teachers must have read in the intermediate book, the given 2 ATP for glycolysis NADH2, FADH2, 3 ATP for Krebs cycle NADH2, decarboxylation NADH2. That's why I had to tell the first 3 number and 2 number. But you should follow our book. Okay? Is it clear? Any doubt?
Yes, sir.
Yes, sir.
No, sir.
Right. So, to be explained about the transport system. Transport system not completed yet. Remember. Hello.
Yes, sir.
Electron transport system not completed yet. I just developed the concept. Okay.
Okay, sir.
Yes,
We
sir.
will discuss about this in next class, electron transport system, because it takes more time. I will explain clearly in next class. And before that, I would like to ask the simple one. Any doubts in for previous unit? Photosynthesis. First, it is clear because these two blocks are very important. Photosynthesis, C3 cycle, C4 cycle, C2 cycle. Already I have sent one of my video about C3 cycle.
Yes, sir.
And photosynthesis reactions explained in on offline class.
Yes, sir.
I think it is clear. Photosynthesis. In photosynthesis, C2 cycle also completed.
Yes, sir.
Right, in plant physical respiration. Should I explain structure of mitochondria?
Okay,
Hello?
sir.
Should I explain? Is it necessary?
Okay.
Sir, I have a doubt, sir. Sir, in glycolysis, one NADH2 produces 2 ATPs, sir. Whereas in Krebs cycle and oxidative decarbonylation, one NADH2 produces 3 ATPs. What is the reason behind
3
it?
or 2.5?
2.5.
Okay.
Why in glycolysis only 2 ATPs, sir, for one NADH2?
I will explain in transport system.
Yes, sir. Okay, sir.
Transport system not completed. That's why I told you.
Okay, sir.
I will explain clearly why only 2 ATP formed in glycolysis. That means 1.5 ATP in glycolysis and only 1.5 ATP in FADH2 oxidation. But
Okay,
other
sir.
2.5. What is the reason? I will explain clearly in electron transport system. Okay?
Okay, sir.
Right, I think you know very well about the mitochondrial structure. It is waste of time to explain, I think.
Yes, sir.
If you want to interest, I will explain.
Yes, sir.
Should I explain now? Mitochondria?
Yes, sir. Yes, please,
Okay.
sir.
Okay, right. Are you able to see this diagram?
Yes, sir.
Right. Mitochondria mainly present in eukaryotic cell only. Eukaryotic cell. Sorry. Discovered by Benda. It is how many membrane? It is covered with how many membrane?
Double membrane, sir.
Double membrane. This outer one. The folding
Inner
one
membrane.
called as?
Cristae.
Inner one. This is called as cristae. Right?
Yes, sir.
Between the outer and inner membrane space called as?
Intermitochondrial space.
Matrix.
Right. Intermembrane space.
Yeah, yes, sir. Intermembranes.
Right. So first let me draw here. Red line. This foldings. Inner membrane shows folding. This foldings called as?
Cristae.
This is called space. And this is called matrix. Between. Matrix, right? So cristae also double membrane. So first in matrix, Krebs cycle takes place. But in cristae layer, this cristae layer, electron transport takes place. This cristae layer, electron transport takes place. Right. Apart from this folding nature, and mitochondria also have the DNA. Actually, where is the DNA?
DNA is in the nucleus, sir.
And other than nucleus?
Mitochondria.
It's present in the matrix, sir.
Other than matrix, mitochondria?
Cytoplasm.
And other than mitochondria, nucleus?
Yes, sir.
Nucleus.
Other than this both, any other cell organelle to contain DNA?
No, sir.
Chloroplast also contain DNA.
Yes.
Remember these two
Yes.
double membraned cell organelles, chloroplast and DNA contains DNA. In general, DNA present in chromosomes of nucleus, right? Is it clear?
Yes, sir.
The simple mitochondria. So no need to, already you know the function of mitochondria. What is the function of mitochondria?
Powerhouse of the cell produces ATP, sir.
Already we have seen how the ATPs are produced.
Yes,
Okay,
sir.
right? Any doubt in glycolysis, oxidative decarboxylation, Krebs cycle?
No, sir.
Clear?
Yes, sir. No doubts.
No. But next class we will continue with electron transport system, and next one, what is the remaining part here of electron transport system? Aerobic respiration, right? Aerobic respiration, and next one,
Anaerobic respiration.
so right, anaerobic respiration. Next one, nitrogen sulfur metabolism. Okay, right, we will meet in next class. Okay, thank you all.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Please follow the notes. You have to prepare your own notes.
Okay, sir.
Right.